Hello, my name is Bartosz Bartmanski, and I will talk about CellNOpt and CARNIVAL software packages in this presentation. First, I will talk about the biological background, and then go into the details of the two tools developed in our lab. First, CellNOpt, and then CARNIVAL. Cell signaling is a fundamental property of cells, be it with its environment or itself. Acquiring a functional understanding of the deregulation of signaling networks in disease would allow us to apply this knowledge to develop novel therapeutics for diseases such as cancer or autoimmune diseases. Our group has developed two tools for understanding of cell signaling, CARNIVAL and CellNOpt. CellNOpt is mainly used on phosphoproteomic data, while CARNIVAL is used on gene expression data. Both of these tools use prior knowledge about the signaling network to gain biological insights. These prior knowledge networks can be obtained from another tool developed at our lab, namely OmniPath. OmniPath integrates over 100 sources of biological knowledge. It started on protein interactions, but things such as expression patterns or ligand receptor resources have been added since then. Now we will focus on CellNOpt and its applications. CellNOpt can build and simulate various models of cell signaling, Boolean, fuzzy, and logic ODE. As shown in the diagram, it takes a prior knowledge network and some additional data to build a model and then simulate it. Logic models are simple, and this makes them very scalable and allows us to model large networks. And they can have continuous variables where we use the logic ODE formalism. Here we have a simple toy example which allows us to easily show the network and the different possible connections between nodes in the network. CellNOpt can assimilate different types of models. The triangles in the plots show data, and the dashed red lines show results from CellNOpt. A more realistic example is shown here, where CellNOpt is used on phosphoproteomics to create cell signaling models for different cell lines that then can be analyzed to determine different effects of different compounds. Here we have the workflow for analyzing the differences across cell lines by feeding the data to CellNOpt, which then builds logic models, which we can then simulate. However, sometimes we have large networks that are built and simulated by CellNOpt, and in those cases, using high-performance computing would help us to quicker and more easily get our analysis. To make use of high-performance computing, as part of PerMedCoE goals, we are improving CellNOpt in the following ways. We are rewriting the R code to C++ for faster execution, and we are incorporating parallelization using MPI. Also, we are making use of hyperheuristic methods, especially ones that benefit from parallelization. To summarize the section, CellNOpt is a collection of open source tools for modeling of cell signaling. It can build and simulate models under different formalisms, such as Boolean logic, constrained fuzzy logic, and logic ODEs. Now we will focus on CARNIVAL. CARNIVAL, which is short for Causal Reasoning for Network Identification using Integer Value Programming, is a software package for the identification of upstream regulatory signaling pathways from downstream gene expression. It uses integer linear programming formalism to determine a solution network based on prior knowledge. Specifically, CARNIVAL takes gene expression data, computes transcription factor and upstream pathways, then maps them on a large signaling network, for example, from OmniPath, and identifies the subnetworks that explain the transcription factors as a function of upstream pathways. As a first study to see how well it works, we applied this to a certain set of cell lines. We then compared the predicted node activities with phosphorylation, which is available for a subset of the nodes. After reconsidering our pipeline, we realized that we could use the RNA to not just input the transcription factors and pathway activities, but use it alongside DNA data to contextualize the networks. Here we have many cell lines to analyze, and this is precisely where we use, where the use of high-performance computing could benefit the analysis. To make use of high-performance computing, we are improving CARNIVAL in the following ways. We have added Gurobi integer linear programming solver, which is the only ILP solver, to our knowledge, which can optimize, which can find a solution network by utilizing multiple cluster nodes. And similarly to CARNIVAL, we are also writing the R code to C++ and utilizing parallelization for MPI, as well as investigating hyperheuristic methods. So in summary, CARNIVAL can be used to infer this signaling network from gene expression data based on integer linear programming optimization to find a solution network. An extension of CARNIVAL called Cosmos can also deal with metabolic networks. Thank you for listening. I look forward to the questions in the Q&A section.